Yeah, thanks for joining us, and thanks PacBio for inviting me to talk about this. So I'm Shane McCarthy. I'm at the University of Cambridge with Richard Durbin's team there, but I work quite closely with the Sanger Institute, and I've been looking at the early data from the Sequel II system that we have up and running here. So from just a sort of, I'm coming from a slightly different background maybe to the other speakers, in that I'm currently involved in a lot of tree of life sequencing projects. So one project being the Vertebrate Genomes Project, which is aiming to get very high quality reference genome assemblies for all vertebrate species. This project has been going on for a little while now, and we're in phase one, where we're creating good genome assemblies for one species per vertebrate order, which is about 260 species or so. And for this first phase, we're focused, we've sort of thrown the kitchen sink at the problem in terms of technologies. So we're, for each ordinal genome, we're doing PacBio, 10x, BioNano, and Hi-C as well. In addition, sort of around the same time as the Vertebrate Genomes Project was sort of kicking off, Sanger was celebrating its 25th anniversary, and there was this project started to sequence 25 genomes from Britain, representing local biodiversity, and that has also led into a larger project, which was announced fairly recently, called the Darwin Tree of Life Project, which is a project to sequence all eukaryotic UK species in the coming years, and that's a collaboration between a number of centres here in the UK. So just to give you an idea of what we're targeting for the Vertebrate Genomes Project, at least, is assemblies which meet this G10K quality metric, which is basically contig N50 greater than a megabase, scaffold N50 greater than 10 megabases, and at least 90% of the genome assigned to chromosomes. And then importantly, also getting base call quality above QV40, and trying to, as much as possible, get diploid haplotypes out of the assembly process. So so we're sort of in the phase of trying to scale up these assemblies if we're going to meet the goals of these projects. So in that context, we're evaluating the 8M system, and we ran a number of different animal samples, non-humans mostly, and I'll just run through some of the samples that we're gonna talk about here. So the first off was the brown trout. This is a nice species that we got from a collaborator in Norway, and it is actually a double haploid sample, which is quite important here because the brown trout genome has a unresolved whole genome duplication, which means some of the genome is in a tetraploid state still. So we previously had CLR data, and we have the other data types from the VGP project for this. We've actually gone through and curated and released an assembly, but we were curious what the CCS data was going to look like for this one. So that's first off. The ploughfish, this is one where we had previously had a CLR assembly, which proved quite difficult. Some fish assemblies, especially that were coming across in the Vertebrate Genomes Project, proving more difficult than a lot of the other species types like birds and etc. So we were curious how CCS was going to perform there. For this Sicilian, which is a large amphibian genome, we were curious how few smart cells we would be able to get to get a good assembly from this. The hawk moth is one which was done for the 25 genomes, and we had existing PacBio data from before and after the switch from version 2 to version 3 chemistry, and so we're curious how CCS would change things there. And for the sterlet, this is another interesting fish where we have an unresolved whole genome duplication, and in this case we have access to a trio where we can do trio binning to separate out the haplotypes. The milkfish is one where we decided, given a lot of the fish in the Vertebrate Genomes Project are proving quite difficult, this looked like a sort of normal, typical fish. We thought we'd, from the same starting material, produce both CCS and CLR data and put it out there as a data set for comparisons. And then lastly, another amphibian where we were trying, it's a large genome, trying to get, see how few smart cells we could do to get a good quality genome assembly out of this. So this is the rundown of our yields, etc., from the various runs. You notice it's not 32 smart cells here. That's because the leads projects that I focused on were sort of half of what we ran here at Sanger. The other half was run as part of the cancer program, so mainly on human cancer samples. But you can see our yields here we were quite happy with them. We had one failure in the hi-fi run here, but then that subsequently ran and gave a good yield here. And for the CLR runs, we did started off a little rocky, but then we've kind of got more consistent towards the end, and we've had runs since this which have been in the high levels here, sort of 80 plus, 80 to 90 to 100 plus gigs. So we're quite happy that after some initial teething, it seems to be running quite consistently in our hands. So I was just going to run through two of these samples here. So I'll run through some results from the sterlet, where we have the trio assembly, and from the milkfish, just to give you an idea of some of the qualities of the data. So for the sterlet, we sequenced 2 smart cells. Both had over 100 gigs in yield per smart cell. It's about a 1.7 gig genome, which means we're quite high coverage in CLR data here, and the read N50s were quite good as well. So as I said, we have a trio here, and we've sequenced the trio, the parents to about 40x on an Illumina HiSeq-X, which means we can apply this nice method from Adam Phillippe's group, which is to use the k-mers in the parental Illumina data to bin the PacBio reads into those which came from the paternal haplotype and those which came from the maternal haplotype. So you can see in the distributions here on the right of the k-mer distributions, this first peak here is the things which are single copy in the one of the parents, so that means those are the hets essentially. The second peak is the things which are 2 copy, so it's the diploid parts of the genome, and this peak over to the right are the things which are 4 copy, so the tetraploid parts of the genome. So using those k-mers unique to either the paternal or the maternal Illumina data, we can assign the PacBio reads, and that worked quite well here. So after trio binning, we had essentially the same amount of data assigned to both the maternal and paternal haplotypes. In the bottom left here is the read distribution. In blue is the read distribution for all the data, and then stacked pretty much on top of each other are the maternal and paternal read distribution. And then here on the right, you can see this is inverse cumulative. So again, blue is all the data, and right on top of each other are the maternal and paternal, and this sort of says we have about 50x coverage at reads of 20 kb or greater. So the assemblies from so assembling each of the parentally binned PacBio data separately, we get quite nice assemblies here. So contig N50 around 6 or 7 megabases. The genome size looks quite accurate, quite spot on. This code here, so if for those not familiar, this is a Busco scores. So this is looking for single copy orthologous genes, which should be a single copy in a particular lineage. So C here is finding if you found those genes and they're complete. And then that's split out into single they're complete and single copy in your assembly, or complete but duplicated in your assembly. And then F is fragmented and M is missing. So for fish genomes, this 92% is quite good, especially at this stage of an assembly, pre illumina polishing or anything like that. And then I guess the interesting thing here is the duplication rate is quite high, and that's because of the tetraploidy. We have a lot of multi-copy genes in this genome. And this plot sort of shows that we are separating out the haplotypes as we expect. So here, each blob represents a contig, and the size of the blob is how many k-mers are in that contig. So it's roughly the size of the contig, proportional to the size of the contig. And then its position on the x or the y-axis is how many of those k-mers are assigned to either paternal or the maternal or paternal unique k-mers. And for a lot of assemblies, you'd get things off the axes here, but for these, we've separated them out quite nicely. Arrow gives quite a similar assembly, slightly lower contig N50, but still quite good from the looks of it. Watchtower here, again, it looks okay, but the genome size for Watchtower here is quite small. So I think you'll see why in the next slide. So here we're doing a CAP spectra plot. So again, this is the k-mer distribution of the, in this case, the 10x reads from the child, from the individual we're assembling. And so things on the left there are single copy, things in the middle peak are things which are 2 copy, and things on the right are 4 copy. And you can see they're color coded by if in the assembly they are actually seen as what copy they're seen in the actual assembly. So ideally we have red here at the single copy peak, all purple here for 2 times at the 2 copy peak, and over here 4 times at the 4 copy peak. And we've roughly seen that here in the Falcon assembly, with some of this green stuff here at the top being stuff which is 3 copy where it should be 2 copy. But that's, I'll come to that in a little bit. Canoe assembly looks quite similar. The Watchtower assembly, you can see what's happened here is that there's a lot of stuff which is, all this black stuff at the bottom is stuff which is essentially 0 copy. It's not seen in the assembly for Watchtower, so it's collapsed it out. And this red stuff here under the what should be all purple are things where it should be 2 copy, but it's 1 copy, so it's been collapsed in the Watchtower assembly. So here the duplication in the Falcon and Canoe is actually due to overlapping contigs, which we can post-process and clean up a bit now. And the Watchtower assembly in this case is collapsing things overly. We're continuing with this assembly. So for the milkfish, we, as I said, we generated CLR and CCS data. So the CCS, this is just what we got for the CCS data. So we ended up with 26x coverage in CCS reads, which was quite good. Mean about 10K, which is expected, and the Q score of a median of about 31. So we assembled with watchtower from this. So we've got an OK config N50, 1.6 meg. And I guess the nice thing about this is you don't have to polish and you still get a good Busco completeness score here of 95%. So Jason Chin got in contact when I put this data out. He has a new, so Jason Chin wrote Falcon initially, and he has a new assembler, Peregrine, which works on hi-fi data. He took that data and tried it out. So he has made an assembly which is looks slightly better in terms of stats than the Watchtower assembly here. So for this data, we have the CLR data set as well, and that's sort of gone through our normal VGP pipeline. The Falcon assembly is sort of roughly 2.5 meg config N50, just doing Falcon on zip, and then we've pushed it through the rest of our pipeline where we've scaffolded and curated. And our current assembly is now a 3.6 meg config N50 in 25 scaffolds at scaffold N50 of 54 meg. Most of it is assigned to 16 chromosomes, and we're quite happy with this. Although we have another version in the pipeline which has a lot higher contiguity, and we're probably going to release this other one soon. So yeah, this is quite a nice test set for people if they want to check out the quality of the CCS data versus the CLR assembly. So the data is available. So VGP is putting out all its data as it's generated. There's a data use policy you can check out here. If you go to vgp.github.io, you can see all the, not just this data, but all the data that the VGP is generating. This here is a summary of where you can get the of the samples that we've run here on the Sequel2 that are VGP and can be released in this manner. So yeah, we're quite happy with the Sequel2s so far. It's a huge leap for these in scaling and affordability for these tree of life genome assembly projects that we're working on. The yields are great, and the vertebrate assemblies are looking quite good. From now we can do them from a single or a handful of smart cells. We have generated some good quality CCS data, but they were still evaluating exactly how those assemblies, how to best use the CCS assemblies for non-human assembly at the moment. So with that, I'll finish, and I want to highlight especially Craig, Karen, and Michelle, who were the people in the lab running the machine and doing extractions and the libraries and everything. And the vertebrate genomes as well. So yeah, thanks for your time, and I guess we're going to head to questions in a sec.